Hello everyone. My poster presentation is on the effects of particle size, solvent type and temperature on the extraction of crude cannabis oil, cannabinoids and terpenes. Introduction includes optimized cannabis processing and extraction improve the yield and efficiency while reducing the operational costs. Particle size reduction or grinding of cannabis biomass improves the extracted yield of crude cannabis oil by damaging the plant cell walls or rupturing the cell walls and increasing the surface area of contact between the biomass and the solvent. The extraction of secondary metabolites including the cannabinoids and terpenes greatly depends on the solvation power of the solvent. Generally organic solvents were employed for the cannabis extraction due to the lipophilic nature of the majority of cannabinoids. Extraction temperature is also influencing the extraction yield and concentration of cannabinoids and terpenes. The main objective of this experiment was to evaluate the effects of particle size, solvent type and different temperatures on the crude cannabis oil yield and on the cannabinoid and terpene concentration using the full factorial design. The three particle size that we used for this study were the coarse, medium and fine. Their sizes were ranging from 2 to 4 millimeters, 0.5 to 2 millimeters and 0.25 to 0.5 millimeters respectively. And the three solvents were ethanol, butanol and hexane. The three temperatures were negative 20 degrees, 4 degrees and room temperature. And this is the flow chart we used for the entire methodology of our experiment. And coming to the results and discussion section, the first part of the results is the effect of particle size on the initial cannabinoid concentration before the extraction. That means after the grinding we sorted out the three different particle sizes and we sent it for the cannabinoid analysis. Results found that the fine particle size is obtaining a higher concentration of the THCA, CBG and CBG, specifically decreasing the particle size from the coarser to the finer, increasing the THCA concentration by 53%, CBGA by 40.2% and CBG by 27.3% respectively. And coming to the second part of the results and discussion, which is the effect of these three parameters on the crude cannabis oil yield and on the cannabinoid concentration. Ethanol combination with the fine sized particles at 4 degrees centigrade is getting the higher yield of crude cannabis oil and higher concentration of THCA as well as the CBG and total terpenes. This is due to ethanol is highly poorer than the butane and hexane. And we also found another interesting result is that irrespective of temperature and particle size, the crude cannabis oil extracts obtained with the three solvents exhibited a different colors. For example, ethanol achieved the darker green, butanol achieved the green, and hexane achieved the yellow color. Coming to the conclusions, particle size reduction of cannabis biomass preserved and improved the cannabinoid concentration during the extraction. There is a positive correlation between the extracted yield with the particle size reduction, and further studies has to be done on finding the precise and optimal particle size for the cannabis extraction and to quantify the effects of different solvents on the extracted chlorophyll concentrations.